tell me about your work at the university in relation to the subject we are talking about, human milk oligosaccharides?
Sure, we've been studying human milk for actually many decades. And we came upon a rather interesting finding that human milk contains undigestible material. That is, mothers are synthesizing molecules that go right through the baby. And when we realized that there was this material, we built an entire platform of analytical chemistry to figure out what it was, how much it varied, and human milk contains an awful lot of oligosaccharides.
So it's carbohydrates we are talking about. We're not talking about proteins which are not digested, IgA, secretory IgA, which comes out. We are talking about carbohydrates. You're saying it's a lot. How much is it?
Interestingly, it's the third most abundant component in milk. There's as much oligosaccharide as protein.
Okay, and is it stable during the period of lactation, or does it change?
There are interesting changes between mothers and during lactation, but in general, it's a relatively constant feature of human milk.
So it's not like the protein, which is very high and then comes down during the period of lactation. It's constant.
Colostrum has elevated levels, so the very first milk has high levels, but then it's relatively constant. We've had mothers who have been lactating for 4 years, and they're still making abundant oligosaccharides.
Now, you detected this bulk of substances which are not absorbed. So what did you do then?
Well, our first inclination was this made no sense. Why would mothers make something that was not digestible by the baby? So we had various ideas, and one of them was, well, maybe bacteria can eat it. And so we isolated the oligosaccharides, and David Mills tested, do bacteria grow on this? And to our surprise, no, they didn't. Most bacteria cannot digest and grow on the bacteria, or on the oligosaccharides.
But they're sugars. They're sugars. They cannot open this, or?
No, they're sugars, but they're held together by stereospecific linkages that babies don't have enzymes to break, nor do bacteria.
So bacteria cannot digest them, babies cannot digest them. What is the use of this?
Well, then David found one bacterium. And this is a strain of bifida bacteria. And for every linkage between the sugars, this bacterium had an enzyme to break it down.
So we are talking about one bug which can digest this sugar? Only one?
Yes, so far we have only discovered one.
And this is a good one. It's not a dangerous one.
It's a remarkably beneficial bacterium, yes.
Okay. So, and it decreases then the amount of this specific bifidobacteria when you have this human milk oligosaccharides?
Yes.
Okay. Now, we know it increases the amount, but why?
Well, that's what science around the world now is struggling to understand. What are all the benefits of this interesting process that came out of evolution, where mothers literally provided food not only for the baby, but for this bacterium. So why would mothers do that? So one of the things that this bacterium does is bind to attachment sites all along the baby's intestine, protecting the baby from pathogens. So pathogen blocking is clearly one strategy.
So this means because you increase the amount of good bacteria, one specific one, they block the bad bacteria.
Yes. So one is it blocks the bad bacteria. The other thing it does is because it feeds this bacterium, it grows and it also produces metabolites. And those metabolites are short-chain fatty acids, and literally the pH goes down. And the lower pH is associated again with protecting the baby from pathogens.
Okay. And that makes sense. Now, it provides protection. This would be more or less a local function in the gut.
Yes.
Does the function go beyond?
Well, so the genius of milk, as it were, is that molecules like this not only have one function, binding to the baby. Two functions, producing acetate. This bacterium also is responsible for producing other metabolites that also nourish the baby. But oligosaccharides are a relatively broad class of molecules, and they actually interact with the host directly. And there's good evidence that they actually form functions to the baby. They signal the baby's immune system.
How do they signal this?
Very early days in science, it looks like the immune system has to decide beneficial, benign, dangerous, pathogenic. And that decision is based on signaling molecules. And by producing molecules that tend to lower the activation of the immune system, then you seem to move towards an immune system that's better for the baby. You also see these molecules signaling to the intestinal cells to start forming the natural barriers between cells that babies have to develop as they mature.
So modulating the immune system, having it at the right level for the baby, also means to be protective against allergy?
The most provocative outcome of the science to date is that yes, that one of the problems with the modern immune system of babies is it's inappropriately activated and reacts to epitopes, molecules that it shouldn't, and forms an allergic reaction.
So now, if somebody from the industry comes to you and says, now I'm convinced what you're saying is true. This is almost a wonder drug in mother's milk. We have a lot of them, a lot of wonder drugs. But this is another one. What should I do? Can I copy it? What is the benefit if I copy it and put it, for example, as supplement or infant formula and give it to an infant?
Ultimately, the goal is to make infant formula as close as possible to human milk.
That's correct, yeah.
And this is a dimension of human milk that is chemical, and yes, it can be produced chemically, and you can reproduce the composition of human milk.
So you add certain components which are crucial of these oligosaccharides, because you cannot add all of them, I assume. This would be very complex.
Ultimately, you'd like to get as close to human milk as possible, but it's going to be stepwise, yes.
Yeah, and so you add them. Now, you have a different environment. It's not breast milk, it's now formula, yeah? Do you expect beneficial effects in this environment?
The basic strategy of nourishing appropriate bacteria seems, at least in part, to be independent of the other aspects of human milk.
So it could work in formula, or it could work as a supplement during mixed feeding?
Yes.
Okay. What would you expect as a clinical outcome or as potential clinical outcome? Less allergies, like in breastfed infants?
The studies to determine all the benefits will have to be done, and this will be built up over years. But one of the things that we anticipate is that this will improve the immune system.
Is there anything from animal models where we can say, okay, we test this? Probably no.
There are data that suggest both reduction in pathogen attack and improvement in protection from allergy. Of course, ultimately, you'd like to see more babies breastfed.
Sure.
Where breastfed fails, breastfeeding is not possible, then this could be a way to improve the trajectory of the immune system of babies.
Bruce, is it only important during the first 2 or 3 months, or would you say this goes beyond the protective effect? For example, when it is added to an infant formula, might last longer, during mixed feeding even?
That's a great question, and there is no clinical evidence at this point, one way or the other. We would speculate on the basis of our results to date that the longer babies can get this benefit, probably the better.
So if I ask you, should I add it to infant formula? If I could do it, I cannot, but companies which have sound research structures can do it. They can test the formula after they produce them. Would you say they should do it?
At this point, the evidence is consistent with this being a valuable component of human milk that would improve formula in moving towards human milk.
Makes sense. Thank you very much.